And the last presentation, sorry, running a bit out of schedule. So the last presentation is by Sophie Roetynck, and it's on the immunogenicity of CHAD63, MVA, ME trap, malaria vectored vaccines.
Thank you. Good afternoon, everyone. So the data I'm going to show you today were generated in the context of a phase 1B clinical trial we completed last August at MRC Unit The Gambia, and it was funded by the different funding body indicated here. So you all know that the development of an effective vaccine is still of high priority in the context of controlling malaria infection, and the different malaria vaccine candidates are categorized according to the life cycle stage at which the targeted antigen is expressed. And to date, the most clinically advanced malaria vaccine are targeting the sporozoite and the liver stages, and among those, one approach that has shown partial efficacy relies on heterologous prime boost vectored vaccination, which has been shown to induce a strong T-cell response, which is believed to play a key role in the protective immune response directed against this stage of the infection. So since many years now, the Jenner Institute in Oxford has been developing a program of viral vectored vaccines, and TRAP is the leading target for such strategy in Oxford. So TRAP stands for thrombospodin-related adhesion protein, is a well-characterized and abundant antigen expressed by liver stage sporozoite, being involved in their infectivity, playing a role in gliding motility, binding to hepatocyte, invasion, and therefore establishment of the infection. So in this heterologous prime boost vaccination approach, two different viral vectors, both containing the ME trap genetic insert, are being given in sequence, and this is to achieve an expansion of the T cells that are specific for trap rather than to the vectors used. So the first vector is the chimpanzee adenovirus 63, and the second one is a modified vaccinia virus Ankara, which is particularly suited for boosting immune responses to an antigen following priming vaccination with another viral vector. And so previous clinical trial of this vaccine in Gambian, Senegalese, Kenyan, and Burkinabe adult and older children shows this vaccine to be safe and highly immunogenic. So this clinical trial here aimed to extend the assessment of safety and immunogenicity of this vaccine in a sample of African infants and neonates. And it was also representing a paradigm shift to South-South collaboration between MRC Unit The Gambia and University of Cheikh Anta Diop in Dakar, Senegal. So we had a scientific officer and a laboratory technician who came to the unit to join the lab team working on this trial. So today I will be just focusing on the secondary objective of the study, which was the assessment of the immunogenicity of CHAD63 MVA trap, as well as of the EPI vaccine when they were given alone or in co-administration with our vaccine candidates. So all the infants received a prime vaccination with CHAD63 ME trapped, followed 8 weeks later by a boost with MVA ME trap. All the infants were categorized into 3 groups according to the age of first vaccination, which was 16, 8, or 1 week of age, and each group consisted of 15 vaccinees and 5 or 10 controls for the youngest age group. So here are represented the frequency of interferon gamma producing cells per million PBMC in response to ME trap as measured by ELISpot in each of the different age group, so and at different time point. So the vaccinees are represented in black while the control are in white, and the geometric means are indicated here in blue for each time point. And as you can see in this population, the vaccine was highly immunogenic, while priming vaccination induced modest T-cell responses. MVA had a boosting effect on this response, which was maintained up to 168 days post-vaccination at post-prime level, and this is irrespective of the age of first vaccination. And we observe 100% responder frequency 1 week post-boost with MVA in all the age group. interferon gamma ELISpot response to TRAP from homologous T996 and heterologous 3D7 Plasmodium falciparum strain were assessed and are represented here for each age group at 4 different time points. So T996 is a vaccine strain, while 3D7 is often used for challenge in efficacy trial, and together they represent the breadth of the genetic variability of the TRAP antigen with 6.1% of amino acid variation. And as you can see, the T cells that are induced by vaccination using TRAP from the T996 strain are highly cross-reactive, suggesting that this vaccine approach should not be too much affected by antigen polymorphism. Using different, in fact, 6 pools of overlapping peptide covering the full length of the TRAP antigen allowed the mapping of the response. So here you have a bar chart representing the mean percentage of the vaccinee responding to each individual peptide pool for each age group, pre-vaccination, 21 days post-priming, and 1 week post-boost with MVA. And as you can see, the breadth of the immune responses as well as the percentage of vaccinee responding to each individual peptide pool increased after each vaccination. And here you have a plot showing individual responses for the peak response 1 week post-boost in each age group. And this data indicates that the peak response was focused towards the 3 first peptide pool covering the N-term region, as well as the last peptide pool corresponding to the C-term end of the TRAP antigen, while response to the central region was rarely observed. So we further characterize this response to TRAP using flow cytometry-based assay, and here the pie chart are representing the proportion of interferon gamma being secreted by CD4 in black and CD8 T cell in gray as a percentage of the total interferon gamma response to TRAP. And overall, interferon gamma was predominantly secreted by CD4 following priming vaccination, while there was an increase in CD8 T cell contribution following boost with MVA. So the peak responses to amitrap 1 week post-boost observed in this population were compared to those previously observed in previous trial using the same vaccine approach, but done in adults and also in older children in the Gambia and Burkina Faso. And due to the observation that lymphocyte count per milliliter of blood can vary with age, we express those responses using spot-forming cells per milliliter of blood unit by integrating the lymphocyte count at this time point. And using this unit, the immunogenicity observed in the 8-week-old group was actually the highest immunogenicity so far observed in African population, and it was almost 3-fold higher than the one observed in UK and Gambian adults, and was comparable to the one previously observed in the same population when this vaccine was being given separately from the EPI vaccine at 10 weeks of age. So anti-TRAP IgG responses to vaccination were also assessed, and so while pre-vaccination levels were all below the limit of detection in all the participants, they increased significantly post-priming vaccination, where 13% of the volunteers, the vaccinees, became seropositive. There was absolutely no significant difference in antibody responses between age group at any time point. Boosting with MVA increased significantly these antibody titers and increased the rate of seropositivity across all age groups to 93%. And these antibody titers remained well above the pre-vaccination level up to 168 days post-vaccination, where 88% of the vaccine remained seropositive. And similar to the T-cell responses, we compared the antibody titers observed in this population to those previously observed in adults and children in the Gambia. And as you can see, titers observed in this trial were significantly higher to those seen in UK and Gambian adults, as well as children in the Gambia aged 2 to 6 years old, but they were significantly lower to those observed in the same population in children when they received this vaccine separately from the EPI vaccine at 10 weeks of age or 5 to 12 months. Having said that, there was absolutely no significant differences in antibody responses to the EPI vaccine between vaccinee and control, although our study was not really powered to detect any significant difference. And this was due to the fact that this unlicensed vaccine was being given to young infants for the first time. So, although I didn't show you that today, our study indicates that this vaccine is quite safe to use and well tolerated in Gambian infants and neonates, and we observed a high level of immunogenicity with a maintenance of the response over time. The highest immunogenicity observed was for the 8-week-old and was comparable to what was previously observed in the same population when this vaccine was being given separately from the EPI vaccine, and was almost 3-fold higher than what was observed in malaria-naive and exposed adults where protection has been observed. The response to trap were broad and highly cross-reactive, and there was a high number of potential epitopes. There was an induction of both CD4 and CD8 T cell, and although I didn't show you that today for a matter of time, there was also an induction of a population of trap-specific monofunctional interferon gamma secreting CD8 T cell that was previously correlated with protection against controlled human malaria infection in naive adults. Although the antibody responses to trap were significantly lower to that previously observed in the same population when the vaccine candidate was being given separately from the EPI vaccine, the antibody responses to the EPI vaccine did not seem to be affected by the co-administration with the vaccine candidate. So overall, given the potential benefit of integrating a new vaccine to an EPI schedule, I think our data provides ground for larger study in pediatric population. And I'm going to stop here with a huge list of the people who contributed one way or another to the success of this trial. Thank you very much.
Questions? So perhaps if there aren't any lingering, if there are any lingering questions from the other presenters, if you would like to ask them, I think that most of the speakers are still here. Okay, thank you very much.